Hello, everyone. Good afternoon, good morning, or good evening, wherever you're watching from. Thank you for joining us today for this seminar, Daylight Insights, So You Want to Measure Daylight. My name is Paul O'Mahony, and I will be moderating this seminar. And of course, this event is hosted by the Daylight Academy. So the Daylight Academy is an international membership organization which connects scientists, architects, and other professionals with a strong interest in daylight-related topics. The Daylight Academy encourages creativity and innovation through interdisciplinarity and disseminating the knowledge amongst specialists and the public. So one of the topics that came up in discussion with the Daylight Academy was daylight measurement. It was apparent in discussions that researchers in different specialties had differing approaches to daylight measurement. We thought it'd be useful to work towards a common framework for daylight measurement, which would enable us to share and learn from each other and broaden our experiences of daylight measurement. To kick this off, we have 4 speakers from a variety of disciplines who are going to share their experiences of daylight measurement with us. Of course, you'll be able to ask your questions. Please do so using the Q&A tab at the bottom of the Zoom screen. The speakers are able to reply directly in the chat, but I will also field your questions during the Q&A session at the end of the seminar. We'll also be able to vote on questions, and the highest scored questions are more likely to be asked during the Q&A. So with introductions out of the way, let's get started with our first speaker. So first up is Professor Brian Norton from Technical University Dublin. So Brian, the screen is yours.
What I'm going to talk about is the measurement of daylight and perhaps how it's not perhaps what it's seen. I come from a perspective which is to do with solar energy in buildings, and daylight in the sense of providing light in buildings and displacing electric lighting. The daylight has a variety of features. It can look at a variety of perspectives, but you've got the solar characteristics itself, spectrum, location on Earth, the sky distribution, and time. It interacts with a set of physical environments, the windows in buildings, the surfaces which reflect within a building, producing heat, interacting with photochemistry, and for example, in photovoltaics, produce electricity. It also interacts with a whole range of organic systems in terms of circadian health, vision, photosynthesis, and these can be mediated visually, and they can be mediated non-visually, for example, through the skin and through the eye. And the physical interactions and the organic interactions themselves interact with each other, so that if you're inside a building, you're receiving the sun through the glazing, through the reflection of the walls to the individual. So these systems are closely coupled. To measurement of daylight it depends what you mean, and I would make the distinction between the measurement of solar radiation and the solar radiation spectrum, and the measure of daylight, which in my world would be the physiological response of the eye to solar radiation. So in that context, when you talk about measuring red daylight, one is not measuring one thing. One actually is measuring 2 things and putting them together. You're measuring the spectrum, and you're measuring the physiological response to that spectrum, and you put it together. And that underlies the definitions that link radiometry and photometry, which I give in this particular slide. And you could say that's it, quite straightforward. There's a relationship between the spectrum, and there's a relationship of luminous efficacy, which gives us the daylight luminous flux for a particular solar radiation intensity. And what I'm going to show in this presentation is, whilst this is true, there's a saying that history is the lies that historians agree on. This is perhaps the standard that people agree on, but doesn't necessarily always reflect reality. So let's look at it in a little bit more detail in terms of what daylight actually comprises. The solar radiation itself has more properties than one might imagine. One's got the intensity, the luminance, the spectrum. But you've also got a temporal pattern across the sky, depending on clouds, their height, their location. You've got a polarization of the light, which is also interacting itself with other surfaces. that comes to us and then interacts with a whole lot of what I would call mediators. And again, depending on where you are in studying daylight, you may have a different view of what the mediators actually are. But at a global level, you've got climate, you've got location, which can be the adjacency of different buildings, but literally latitude and longitude and solar motion. You've got vegetation, and that's the environment. Then from a building's perspective, that light is going to come through apertures, it's going to interact with materials, it's going to be subject to control. That might be fixed controls like fixed shading devices on the external side of the building, it might be blinds. It's going to interact with the form of the building, it's going to interact with the heating and cooling system in a variety of ways, in that windows will be adjusted for if the building's overheated, blinds and curtains may be closed to retain heat. Then, having gone through all those processes, it's going to interact with the eyes to provide visual stimulus. It's going to interact with the skin and have a variety of interactions there. But it's also going to interact even with the eyes in a non-visual sense to give cues to circadian health and signals to behavior. And those will vary depending on the environment of the person, their age, and a whole range of other factors. So immediately we see it's rather more complicated. And I've drawn these as kind of circles, but then got this kind of pyramid here. And I've drawn this pyramid really to emulate a sort of hierarchy of needs, because all these things are meeting a whole variety of needs that we have for daylight. There's a whole set of physiological ones that it's actually providing biorhythmic health, it's providing visual acuity, comfort, warmth, safety. Okay, there's a whole lot of psychological needs that are there in terms of privacy, exterior views, so quality, but also it has a whole set of cultural from aesthetics, but also as I say, I work in the area of displacing electric lighting, a whole set of economic benefits. And economics is actually a cultural artifact, so there are a whole set of cultural benefits, and these are not so easily disentangled. So daylight and its perception, its measurement, and its value has a whole degree of complexity associated with it. If you looked at that from a different perspective again, and this is a diagram taken by paper from Boyce et al. This is looking at the daylight in terms of how it affects visual performance in buildings. And you look at the lighting and the task. You start from that perspective. You're looking at the visual area, looking at the luminance, looking at the color contrast in there, you're looking at the retinal image density, etc. A whole lot of very detailed aspects of the eye influence how the eye, even from the context of visual performance, and here we're not looking at necessary non-visual parts of the eye, not looking at whether this is in light or dark conditions, but just generally looking at visual performance. A whole set of things come into there. They are in turn influenced by things to do with the motor performance of the eye, whether one wears spectacles or not, fatigue, etc., and a whole set of human performance factors. So what constitutes effective daylight is not simply a function of those two measurements taken together, but is very contextually driven. Interesting, just on the cultural question there, a lot of daylight studies are done on latitudes and in conditions that are essentially temperate climates, with solar motions that are above the tropics, and whether those assumptions are actually applicable into other climatic conditions and indeed cultural environments and how those interact is in itself an interesting question. And indeed, if you start taking that analysis, the solar motion on the right hand side of this figure, as you see it, varies heavily with latitude, and in fact, it's only at the equator that the sun will be, for example, directly overhead. And it does mean that at high latitudes, both in the northern and southern hemisphere, in summer the sun is actually not rising in the east; it's rising in the northeast and setting rising behind you and setting behind you, and it's actually quite high in the day. So when you're coming to looking due east, so quite complex bathe of the sun across the sky. That means the sun is every day going through different thicknesses of atmosphere, and the solar spectrum changes with different thicknesses of atmosphere. Now, other speakers can talk about this in a lot more detail. But the key thing is that at specific locations, let me just go back there. My slide went forward. At specific locations, the luminous efficacy is going to vary considerably over the day as you encounter different air masses, different aerosols, water vapors, type, extent, height of clouds, etc. And real luminous efficacies are different from the diffuse solar component, and in daylighting work historically, the diffuse solar component was all that was actually looked at, and also they'll be different for the direct solar component as well, and that will be particularly affected with solar altitude because it's directly a function of air mass. So again, the spectrum is not one thing, it is changing over the day and giving you different luminous efficacies. Those and just to illustrate that point, you can see the kind of different spectrums that you see and the kind of devices that would attach themselves to different spectrums, and how skies then actually appear as spectra. So it's actually quite complex phenomena. In buildings, that is also, if you're in a warm climate, in a cold climate, I'll start with that one, you're seeking both to emit solar heat gain and daylight, and use that as part of the passive solar heating of the building. In a warm climate, you're designing glazing systems with coatings, or using glazing systems with coatings, that only would allow in the daylight part of the spectrum and reject the heat. So how much spectrum is emitted and the associated phenomena that come with it are affected by the choice of glazing system. And indeed, that then maps on, as you can see here, to the particular spectrum, because there's not just one visual spectrum. There is a visual spectrum for light conditions, a visual response for light conditions, a visual response for dark conditions, and indeed a totally separate non-visual response that controls the circadian health. And other people are going to talk about this a little later. So the question would be, what spectrum do you match onto what and when? And for what purpose is one doing that? Now, various standards have been developed. So there is a standard which underlines lighting phenomena, which defines standard spectrum, defines standard responses, and that's fine if you look at standard conditions. But the reality is that actual conditions are not those that are applied to those standard conditions. And if you've got situations where that's not the case, one needs to do something different. So, for example, in the built environment, to take account that you've got variations of intensity, spectrum, and anisotropy of the sky over the day, one's moved from a situation where there was a percentage of the luminance cast by the overcast sky was what you wanted to calculate on a working plane in the building, which is shown in the grey line to the right of the diagram on the right here. And that was how one did a daylight calculation. And for example, if you'd achieved 5% of the external daylight, so a 5% daylight factor, you had and you were looking at the overcast conditions as the key driver there. Modern calculations have moved away from that to actually have a calculation of daylight which is computationally intensive, but actually is climate based and based on the real climate factors at that point in time. So you're looking at both daylight, you also look at skylight, you're calculating the two together, and you're looking at the total effect that actually happens in terms of the daylights available, and designing with that spatially over time, and looking at how the lighting conditions can be met from daylight in a holistic way using real climatic data. So that has happened in the built environment and has moved away from a static response to a dynamic response that includes all factors of daylight. Now, the question would be, does that have to happen in other areas? And I would say probably there needs to be further work here. If you look at the non-visual response of the eye, work has shown that actually that is not even a static thing in itself. It varies with age. Now, if it varied with age in a systematic way, so that this graph just declined in magnitude in a systematic way, that might not be an issue. But in fact, the peak is also changing. So that will have an effect when you multiply this spectrum with this response to a particular spectrum. The response is more complex than a simply thing that could be weighted by a factor to allow for age. Also, spectrum vary with angle. In other words, the actual perception of the angle of view varies. This is a response to glare, and glare is where it's directed into the eye. There's more tolerant at the edge of the eye. So what plane are we measuring on? And traditionally, things will be measured on a working plane. In fact, daylight studies in buildings would have looked at daylight on the horizontal working plane, on the received surface, would be where the daylight would have been measured, not necessarily at the eye. So where it's actually measured becomes important, and there are clear, obviously, angular effects in terms of what's received, in terms of the angle, the reflection through the walls, etc., but also clear implications for the eye itself. And talking about what's received, the transmission through glazing is quite interesting. So we've seen sky conditions and see how they vary, but actually, if you look at glazing, and this is a transmission of a particular glazing system with the clearness indices of the sky. And whilst you have, if you've got above a certain clearness, you've got a variation of transmissions with the angle of the sun. So these are clear conditions. Below a certain cloudiness, that ceases to be in effect. So the nature of the glazing will create angular effects above at certain conditions, but won't create them at other conditions. And in a real internal environment, that will interact with the angular response of the eye and the physiological response of the eye to the spectrum to give you quite different effects from what you see. And also the responses vary with the receptor. And again, these are standard responses, but these are also measured variables, and they're measured for particular individuals in particular places. And more information about what these are, but also their magnitudes and how they relate to one another is needed. So it's a very complex picture in terms of the actual response. So if you look at this, so this table is deliberately incomplete before I get the comment that I've presented an incomplete table. But what it's an attempt to do is to try and relate this against a few disciplines. And I suppose what I'm presenting here is an initial attempt to something which may become a very large table, which tries to relate the different ways that daylight is looked at. And I've picked a few domains here of, say, physics, and you've got a whole range of parameters for flux, intensity, photons, the directness of the solar radiation, its polarization, for example, and that essentially is the spectrum. And then you've got physiological, people, and in here you've got visual phenomena, the luminance, the visible flux of visual luminance intensity, for example, and you've got non-visible, circadian luminance flux, and circadian intensity, and they can be related to each other depending on what response one's multiplying against the spectrum. Okay, but then we look down a bit further. If you look at that for example, there'll be a photon count, and if you're looking at photosynthesis, and we'll be hearing about photosynthesis later, photon count is actually used. Okay, but it's not used in physiological work as far as I know, and again I stand corrected here. You could have radiances and flux and things for the photosynthetic area, but how do they relate to photon-based measurements? Coming into there, ideas within, for example, buildings would be idea of daylight autonomy. How much can you daylight the building without electric lighting? Are there similar kinds of ideas that might go across from photosynthesis or from, for example, the physics idea, and also things around the usefulness of daylight? And again, there are phenomena in there in visual photometry, and there are similar ideas in photosynthesis. Now, there may be things to fill in all these boxes. I don't know. And there are probably a lot further boxes. You can look, for example, at photochemistry and many other subjects going across here, but begin to look at the relationships. So possibly there might be one thing to do is develop these kind of tables, and maybe not simply to say that this is how it's done, but why is it done in one discipline, which is essentially the similar underlying phenomena, and not done in another discipline, and whether there is an insight that can be gained from doing that. For example, there are many phenomena that can be explained by wave theories of light, so they can be explained by photon theories of light, corpuscular theories of light. Are both needed to be able to explain particular phenomena in particular systems. So the challenge, the challenge is that there are a huge variety of techniques and units to measure daylight. There are divergent weightings applied depending on the systems examined, but within the boundaries of particular disciplines, these are well understood, but across boundaries, not so. So the challenge is how to translate between disciplines as varied as chronobiology, photosynthesis, photochemistry, meteorology indeed, lighting engineering, architecture, and many, 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 many others. How do you translate information there? And one project that is going to be doing this as part of an exercise within the Daylight Academy is the Daylight House project, to try and develop a shared understanding of how the quantities are derived, how, for example, common computational techniques are used to understand how these can be correlated, decorrelated, try to understand effects that are not apparent because of the metrics used. And also, and I haven't really gone into this in great detail here, where things might be codependent on multiple inputs, different distinct parts of the spectrum, or perhaps ambient temperature, so that metrics and concepts can be used outside the original discipline, and particularly in systems where perhaps different concepts are present in the same environment, one can get a mutual understanding. And so ultimately, that, for example, one can take examples from physiological studies of how the human brain responds to light and take them into things like the design of buildings. Thank you very much. Happy to answer questions later on.
That was brilliant. Thanks, Brian. That's a really good introduction to the topic. As I mentioned before, you can put questions in the Q&A chat, and we will field questions to the speakers at the end. So next on the agenda is Manuel Spitschan from the University of Oxford. Manuel, the screen is yours.
Hello, can you see my PowerPoint? Just confirming.
Yeah, that looks great.
Perfect, great. Well, thanks, Paul, and thanks to the Daylight Academy for inviting me. It's a really great pleasure to be part of this interdisciplinary group, and I really hope that what we're talking about here will lead to ongoing discussions and really kind of stimulate our thinking and how we can tie the different concepts that we have developed in our relative disciplines together to really derive an integrated understanding of daylight, and effectively, in a way, also then derive new research questions and new translational approaches to maximizing daylight. I should say a little bit about my background. I'm a vision scientist by training. I've since then moved into the field of chronobiology, and really my goal is to integrate between these two disciplines a little bit the knowledge that we have about how light affects visual and non-visual responses to light. So just to get started, this is probably for some of us a very familiar diagram, for others maybe something they've last seen in high school biology. I should say my framework really in thinking about the effects of light, including daylight, is really a retinally referenced one. So the way I understand the effects of daylight on physiology and behavior necessarily take place in a framework that makes reference to the human photoreceptors that Brian has already introduced. And of course, the way this works is you have different photoreceptors in the back of the eye and the retina. You have the cones, the rods, and the IPIGCs. Until about 20 years ago, we thought, or the field thought, that cones and rods are the only photoreceptors in the retina, until the discovery of another class of photoreceptors. And these are the intrinsically photosensitive retinal ganglion cells that express a specific photopigment that is called melanopsin. Melanopsin is short wavelength sensitive, and it is quite distinct from the other photoreceptors. Of course, the retina processing in the brain doesn't end in the retina. It goes to various targets. And I think the one that is most studied by vision scientists is, of course, the projection from the retina back to the back of the brain to the visual cortex. And so, of course, then if your eye is exposed to specific light, that will then cause specific signals to arise in the retina, and then thereon. We've seen this diagram before. These are the spectral sensitivities of the human photoreceptors. What's really salient here to me is that they all have different peak spectral sensitivity, so their wavelength preference is different, but they're all very overlapping. And I think for purposes of examining what different photoreceptors do, this poses an interesting question, because there will be no single wavelength light that's going to only activate one class of photoreceptors. And of course, I think there's something to be said here about the range that these photoreceptors cover. And of course, if you want to argue in some evolutionary framework, you probably might pick up an argument to say, well, there's a reason for why the photoreceptors have such distinct wavelengths sensitivities. Now, what happens when we are exposed to a light, as I mentioned, it triggers a set of signals in the human retina, and generally we make distinctions between visual and non-visual processes. Of course, it all goes through the retina, right? So I think maybe a more nuanced way of talking about this would be to talk about the retinally referenced or retinally mediated effects of light, but generally we're talking about different types of outputs. And I mentioned before, visual cortex is one area that's activated, and it's assumed to, there's converging and a lot of evidence really that primary visual cortex and projections from primary visual cortex underlie our visual processing of color, space, and motion, as well as anything really that we associate with the term visual perception. Now, in contrast to that, there's another pathway that connects the retina to the what's called the suprachiasmatic nucleus. And the effects that are mediated by this pathway are what's generally what are generally called the non-visual effects of light. And this includes a range of effects, including the suppression of the hormone melatonin by light in the evening and at night, the shifting of circadian phase in response to light, and as well modulation of alertness. And there's some other functions or physiological parameters that are also modified by light. Of course, what we want to do in practice as vision and circadian scientists is understand a little bit how the human response depends on different kinds of inputs. And one realization that is quite striking is, of course, that we are exposed to different kinds of spectra during the day and at night. And I'm just showing you here 3 types of daylight, lights that we would consider daylight, and they differ by the amount of effectively short wavelength or long wavelength content. The way you would describe these daylights generally is in terms of the correlated color temperature. And so this corresponds to the temperature of a black body radiator that has the same color as the daylight in question. These are normalized spectra, right? But these are basically all plausible daylights. And when we're talking about D65 daylight, that's a relatively neutral daylight. It has a correlated color temperature of 6,500 Kelvin. Of course, with the advent of electric lighting, we are not only exposed to daylight or variants of daylight, we're also exposed to artificial light. And so now there's a huge spectral diversity in a way of things that make light. And I'm just showing you 3 examples here: white light from an LED, fluorescent light, and the standard illuminant, incandescent. The interesting thing here is that they all have the same luminous flux. They all have the same lumen output. But of course, they have different spectra. And what is really important here is that the human visual system, of course, doesn't see spectra. It sees everything collapsed into the 5 photoreceptor space that we have in the human eye. Another thing that's really important is that daylight exhibits what we call statistical regularity. Of course, as I've kind of foreshadowed here in the previous slide, there are ways of looking at daylight and ways that daylight can look like. And in the 1960s, there was a really, really important effort done by various people who are really the giants of color science, as it were, looking at the properties of daylight and how daylight varies and how it can be described in an efficient way. What they found is that daylight across a range of different colors can be described by mean and 2 components. And so basically what we can do is we take a mean daylight, which could be the D65 spectrum, and we just add 2 components to it to basically create daylight of any correlated color temperature. And this is really an important insight, is that daylight is statistically regular. We can, of course, then project this into what is called the CRE chromaticity diagram, which is basically plotting the color appearance of a given light in a human-centric way. This is what's called the horseshoe diagram as well. You might have come across this, but basically what we can do is every spectrum that exists, we can calculate its chromaticity coordinates to. And so the fact that daylight is statistically regular basically means that all daylights will lie on what's called the daylight locus. And so that is basically a line or a curved line in the chromaticity diagram. And so again, of all the possible ways that natural illumination could occur, daylight is relatively constrained. One of the things that I was interested in a couple of years ago was understanding, well, okay, we talk a lot about daylight, and I think we have a lot of models and a lot of ways of thinking about daylight. But I was really interested in understanding what happens when we're moving outside of daylight into the twilight zone. And so what I did was taking measurements of natural illumination in rural Pennsylvania, where there was no or relatively little artificial illumination. And what's really interesting is that you have, as you're progressing from daytime light exposure through twilight, this very, very strong short wavelength boost of light during twilight. And so this starts very much during civil twilight, then moves to nautical twilight, and you just have a very, very blue sky in twilight. And of course, physiologically, the availability of light is then also linked to the types of photoreceptors that are active during a given light intensity. And so twilight is typically associated with what's called the mesopic zone, or the mesopic range of vision, in which both rods and the cones are active. These were relatively privileged measurements in a way, because if you typically take these types of measurements, and this is in the city of Philadelphia, what you just end up with at some point during nautical twilight is basically sodium high pressure lamps or some other artificial illumination that just dominates the night sky. And so you can see here these peaky spectra correspond to basically the street light and whatever light was then reflected from the sky. Of course, we can look at this not only in terms of spectrum, but also in terms of the color. And of course, you can then see that basically, so these are isoluminant representations of this. As the sun is going down, the sky gets bluer and bluer in the rural location, and at some point turns orange in the city locations. We then did a little bit more modeling of these types of data, and what we did was specifically expanding the daylight model to incorporate also the ability to model then twilight components in the night sky. When we're looking across a range of spectra, so moving on from daylight, what is really interesting is then to examine how the melanopsin signal, right? So the signal that's available to the IPRGCs, that's underlying non-visual functions, how that varies with different light sources. And here, what I'm showing you is basically the melanopic irradiance of a given spectrum at 100 lux, right? So this is a hypothetical exercise. I take a spectrum, I set it to be 100 lux, have an illuminance of 100 lux, and I ask, what is the melanopic irradiance that you will get at this illuminance? And there's various hypothetical or various spectra that I examined here. And so interestingly, on average, the daylight spectra here, of course, have the highest melanopic irradiance at 100 lux. And one of the intuitions for this is basically that the daylight spectra are broadband sources. So they can pack much more photons in that will be melanopically or relevant to the melanopsin system. We can also look at the same data in a different way, just plotting the correlated color temperature against the melanopic irradiance. And you can see here, again, somewhat the daylight locus. And most of these spectra that we have in this database are falling below the daylight locus, which I think is a very, very interesting finding. Now, the thing that we mostly care about, I think, in circadian science is understanding a little bit of the time course of light exposure. And so we developed a little bit this hypothetical concept called the spectral diet, which effectively relates the time course of light exposures over the 24-hour day. It's not very easy to measure, and I'll come to that in a second. Here's some pilot data that were collected a couple of years ago, and you can easily distinguish different types of data or different types of spectra in these data already. And ultimately, of course, what we care about is relating these to physiological measures. Coming to the question of practical measurements, I think one way of thinking about the ways that we can measure light for circadian purposes is in terms of two dimensions that are relatively easy to understand. One is the spectral resolution, right? So the amount of spectrum that I can really capture with a sensor or with a given measurement technique. And the other one is the temporal resolution. And I'm just going to populate this kind of 2D space with a couple of instruments. With the spectrometer, I could measure with high spectral resolution, but generally with very low temporal resolution. Of course, you can find also ActiWatches with lux sensors where you can have a very high temporal resolution, but very low spectral resolution. And the kind of, I guess, quadrant or the kind of the part of the space that I think the field is moving towards, or that becomes very, very interesting and important, is the space of measuring at both high temporal resolution and high spectral resolution. And there's various devices that have been developed by various research groups around the world that aim to exactly do that. In my own lab, I've done a couple of experiments. One is specifically ongoing. This is work led by Raphael Lazar, looking at measuring aspects of real-world light exposure and relating it to a physiological output that is relatively instantaneous, and that's the pupil size. One of the problems that we see is that we can, of course, look at parametric models or parametric measurements of pupil size under different lighting conditions, but that doesn't necessarily tell us what the pupil responds to or the pupil size is under a range of real-world spectrum. So we developed this conjoint measurement device to examine effectively spectrum and pupil size in the near corneal plane in ambulatory measurements. These are just some data that we've recently collected. effectively showing, so on the x-axis here, the melanopic irradiance, on the y-axis, the pupil diameter, quite noisy data. And I think this speaks a little bit to this question of how can you, how easy is it to make real world measurements? But certainly on all of these cases, you see a clear non-monotonic relationship between pupil size and the melanopic irradiance. We can also express the data in a way that basically looks at the spectral properties. And so in general, I think this is something that's really interesting when we're plotting photopic illuminance against the melanopic irradiance. You have a lot of data points that just fall along the diagonal. These individual islands of points that are not on the diagonal are measurements that we took under narrowband illumination in the lab, but effectively everything that's along the diagonal really corresponds to the measurements that we see in the real world. So there's a strong correlation between melanopic and photopic signals. Just one thing that I've recently developed is an open access platform that hopefully might be useful for some people in the audience, where we can calculate various quantities based on light and lighting. I just maybe can switch to this quick window here. This is an example spectrum. It's not daylight, it's actually moonlight. I took a measurement of the recent supermoon and just fed it into this. And you can beautifully see how, in fact, this looks very much like what you would expect, which is some version of sunlight. Finally, as part of the Daylight Academy, part of a group of Mirjam Münch, Deborah Skeen, Katharina Wolf, and Martine Knoop, where we've been looking at developing a human light exposure database, where the idea is to really aggregate data from different geographic location and contexts and put them together in a common resource. And I think this is something that's really, to me, a key development in a way, because I think we can then harness in a way the power of big data, specifically when it comes to human daylight exposure. I think my time is up. Thank you so much for listening. I'll be happy to take any questions after the talk.
Thanks, Manuel. That was great. Nice jaunt through the twilight zone of daylight. So a reminder, you can ask your questions in the chat and they will be asked to the speakers at the end of the session. So next on the agenda, we have Professor Anne Webb from the University of Manchester. The screen is yours, Anne.
Okay, thank you. Is that visible to everybody?
Yes, that is. If you just start the slideshow, that'd be great.
There we go. Okay, well, thank you for the invitation to speak here. I'm going to be talking to you about solar ultraviolet radiation, which isn't technically light, but it is part of the solar spectrum. I work in the Centre for Atmospheric Sciences at the University of Manchester. So my background is in measurements from the atmospheric, climatological, meteorological point of view. But I also work a lot with the effects of ultraviolet radiation. And so looking at its impact on mainly human health, but also other forms of life. So I thank our previous two speakers for setting me up so well. I think you'll see a lot of similarities or links between what I'm going to say and what they've already spoken to you about. So let's start off with why we want to measure ultraviolet radiation. And really the big impetus for it came from the Antarctic ozone hole. So there were very few measurements of ultraviolet radiation. I don't think people really took a great deal of notice of it until the 1980s, when the Antarctic ozone hole was discovered. And ozone protects us from the shortwave ultraviolet radiation in sunlight. Shortwave radiation is damaging. It's damaging to our DNA and damaging to the DNA of all life forms. And it's the ozone in the stratosphere that stops those very shortest wavelengths reaching the Earth's surface. So it was really the development of the ozone layer that enabled life to crawl out of the water and start to live on land and be exposed to all the solar radiation. So along came the Antarctic ozone hole, and theory and indeed practice says that where we have less ozone, we get a lot more ultraviolet radiation at the Earth's surface. So this caused a great deal of consternation. What would happen if we got lots and lots more ultraviolet radiation at the surface? In terms of the direct, immediate impact on humans, we'd probably get a lot more sunburn. As you know, that's a risk factor for skin cancer. And so the immediate message became, don't do it. First of all, don't deplete the ozone layer anymore. Let's stop doing that. And there was a very successful Montreal Protocol international agreement to stop using chlorofluorocarbons. But also the public health message became, be very conscious of ultraviolet radiation, don't go out in sunlight, don't get a sunburn, which remains good public health advice. But what it led to was a sudden increase in the desire to measure ultraviolet radiation, because there was very little detail about how much UV there was at the Earth's surface. And so if we don't know what's here now, how can we tell if it's increasing and the sort of damage that it might do? So here are some of the biological effects that are due to ultraviolet radiation. It does, of course, cause sunburn, which is a risk factor for skin cancer in humans. It does have a positive effect. Your skin makes vitamin D when exposed to ultraviolet radiation. And that's something that I do a lot of my work on, is on the positive benefits of ultraviolet radiation. But there is that tension between getting enough for your vitamin D and not too much so that you get a sunburn. But as I've said, UV does impact on DNA, so it has impacts on plants. It tends to reduce growth and yield and has an impact on the photosynthetic system in plants. But it can also affect things like stomatal opening. And there's some fairly recent work that I've done with people in Bristol looking at the balance between the UVB, the shortest wavelengths of UV radiation at the Earth's surface, and the slightly longer wavelength UVA. And it seems that the ratio between that UVB and UVA might act as a trigger for stomatal opening and closing in plants. So a little bit like a kind of stomatal circadian rhythm in plants. UV also affects aquatic systems. UV is transmitted through clear waters. And so it does get down to some depth when you get out into the clear ocean waters, maybe not quite so much right at the beach. But it affects zooplankton and phytoplankton, and they're both really quite sensitive to UV radiation. And so it's impacting both on photosynthesis within the aquatic systems and on the very bottom of the aquatic food chain. So UV is important not just for our human health, but also for a lot of other life forms. So I'm now going to show you a little bit about the sort of history of what went on once the Antarctic ozone hole had been identified. And we started to think about measuring solar radiation. So the main challenge with measuring solar radiation is, in fact, the solar spectrum itself. Let's see if I can get myself a pointer here. There we go. So we're dealing with this tiny bit of radiation at the short wavelength end of the solar spectrum. So you've seen this picture before already today, but this is the extraterrestrial spectrum in yellow, and then there's a lot of absorption that goes on in the atmosphere, and then at the ground we get this red spectrum. And you'll see here that ozone is absorbing the short wavelength end of the solar spectrum and diminishing what we receive at the ground. We are interested from a human health perspective in both the good and the bad aspects of solar UV radiation. So erythema, this is sunburn. This is the response spectrum of human skin for sunburn, and you can see it's at a maximum in the UVB, as is that for the positive benefit of vitamin D synthesis, again, a maximum in the UVB. And the DNA action spectrum is very similar to these, but drops off even more sharply in the short wavelengths. So we end up with, if I put this on a log scale, this solar spectrum increasing by orders of magnitude across a very short wavelength range, while the biological impacts are decreasing just as steeply across, again, a fairly short wavelength range. So when we're measuring UV radiation, it's really important, if we're looking at biological effects, to understand this and to be able to account for this, to be able to measure this very steep wavelength drop off with wavelength. And so the gold standard for doing this is to have a double monochromator spectroradiometer and a photomultiplier tube. So it's very sensitive, it has a high wavelength resolution so that you can really pick out this part of the spectrum that's so important for the biological effects. But these things are big and they're expensive and they're very sensitive. They basically need to babysit them if you're going to get good results. So as Manuel alluded to, we also can use much smaller instruments, single monochromators, that then have a diode array detector of some sort. And these are much quicker. You can measure the whole spectrum at one go instead of having to sample a wavelength at a time with the double monochromators. But they tend not to be as sensitive. You tend to lose some of the sensitivity down in this part of the spectrum if you're trying to measure solar radiation. And because they're only single monochromators, then you can get a lot of stray light. One of the problems is that you've got so much more radiation in a very close part of the spectrum. And so if you're not careful, you get light from here being detected as light from here. And that can lead to misunderstandings of what's happening. As you might imagine, you have to have very careful wavelength alignment across this part of the spectrum, because if you shift things by only a little bit, then you're measuring in error by several orders of magnitude. And we need to know what the optical input of our, or the response of our optical input is. In meteorology, we measure the radiation on a flat surface. That's the sort of standard measurement. We might also measure direct radiation, so that's the direct beam coming from the sun, at which point you have to have an input optic that looks directly at the sun and tracks the sun so that you're only really measuring the solar disk. So those are my standards, if you like. But you might also want to measure the radiation incident on a point. So radiation coming from the full 4 pi around a small point. And that, in my world, would be important for atmospheric chemistry, for example. And then, of course, you need to calibrate all of this. So my basic instrument is a double monochromator with a photomultiplier tube, but that's difficult to employ in a lot of places. So there are other measurements. Broadband measurements and dosimetry are used a lot in public health. So broadband radiometers for this application are designed to have a response like the human skin for erythema. And so the measurement that you get from these is directly related to how your skin would respond in terms of sunburn. So they're very useful for public health messaging, but they do have to be carefully calibrated, because as the solar spectrum changes, and it changes quite a lot across the UV, depending on how much ozone is in the atmosphere and on the solar elevation, then you have to do a careful calibration. And dosimetry is used for measuring exposure to people in everyday life, because of course we don't get the same exposure as the flat horizontal surface. And so you can see here, this is some exposure from dosimetry badges, which also have an erythemal response, and it was measured across a whole range of adolescents. So here we can see that, as any of you who have adolescents know, a lot of them like to stay in bed at weekends and don't get any exposure, and yet some of them go out and get a lot of exposure. But it's only about 1%, the median's only about 1% of the ambient. This erythemally weighted UV is also used in the UV index, which you can find in weather forecasts. And this is actually the weather forecast from, I think this was Holmfirth in Yorkshire on Friday, when it was a beautiful sunny day. And you can see that we're getting a UV index of 6. This is just a sort of normalized measurement of that sunburning, erythemally effective UV. In the UK, we never get a UV index more than about 8, but of course, as you go further towards the equator, it can become much more extreme. But it allows us to estimate how strong the sun is in the UK and compare that with where we might go on holiday, for example. And I just went to have a look and check whether this forecast was right. And indeed, the UV index did get to just about 6 on Friday. So these are other measurements made very much in terms of UV index for public health and for dosimetry for our research purposes. So back in 1985, when the Antarctic ozone hole paper was published, there were very few measurements. And since then, many individuals or national efforts have been made to improve solar measurements, solar UV measurements. But to be able to compare across the climate and to be able to look at the whole globe, which was really important when we're looking at ozone depletion or potential ozone depletion across the whole globe, it was really important to know that the UV measurements being made in one place were directly comparable with those being made anywhere else. And people were using a lot of different instruments. There weren't many that were designed for use outdoors. Only the Robertson-Berger broadband meter, which was being used in the US and Australia to look at sunburn and skin cancer, and the Brewer spectrophotometer, which was primarily developed for ozone measurement, but does make some spectral UV measurements. So many people, myself included, started to use modified laboratory instruments for outdoor use or to build their own instruments. And the question then was, are they all measuring the same thing? So we were fortunate to get a lot of European funding in the early 90s to bring everybody together and put the instruments side by side and see if they were indeed comparable. And the answer back in 1990 was no, I'm afraid not really. So there was a great deal of work went on over really quite a short time to improve these spectral UV measurements, initially with a sort of atmospheric climatological input towards the whole ozone hole problem, but also leading on from that for public health. And it led to, after 1997, things were getting much better then, led to the development of a travelling calibration system. Again, it was developed with EU funding and was later adopted by the Swiss at the World Radiation Centre in Davos, who now operate it on behalf of the World Meteorological Organization, first as a regional, a European calibration centre, and then as a global calibration centre, so that we can link up all these measurements of solar UV radiation made around the world and know that we're comparing like with like. So the measurements, the basic climatological measurements are made in sites like this. So this is one of the sites where my instruments are. This is actually the meteorological site at Reading University.
Right, just 2 minutes left, Anne.
Okay, and there was a lot of work done to process the data so that, again, we're comparing like with like. Everything's on the same wavelength scale. Everything's got a uniform slit width. And this is some data courtesy of Julian Grosvenor at the World Radiation Centre, showing how you can get good comparisons like this one, where all the wavelengths across all the times of day and all the solar zenith angles are very close. So that's a good result. This is one that's not doing quite so well. There are different wavelength comparisons and things are drifting with time of day. If we'd got this back in 1990, we would have thought it was amazing. The scale there was from about 0.5 to 1.5 and not all the instruments fit on that. So I think UV measurements have come a long way globally over the past few decades, with a lot of that work being done in the 90s. Just a note that with artificial UV, that's also used for experiments, as I'm sure artificial light is in many trials. And again, it's really important to measure that carefully and also make sure with artificial lighting that you're going to the shortest wavelengths and not missing something that might be very effective. So pitfalls in UV measurement, and I'm sure in the rest of daylight measurement, is in not understanding your instrument. You do need to understand exactly what you're measuring, otherwise it can lead to misinterpretation of the data. When you're reporting it, when you're writing papers, and this is a plea from someone who's trying to do a systematic review, please give all the details, report what you're measuring, whether it's weighted or unweighted radiation, what your field of view is, how it was calibrated, so on and so forth. So it is really important to make sure your calibration is good. And if at all possible, make spectral measurements because they are so versatile, but they are challenging to make. And sometimes it's easier to use broadband measurements. So a lux meter if it's daylight, a broadband erythemometer if it's me. And just to link back to what Manuel was talking about, we've heard all about circadian rhythms. This is some spectral UV work that we did with Rob Lucas and his team in Manchester. They were working with mice, and mice have a UV opsin, so they are sensitive to UV radiation. And so they wanted to know how the spectrum changed, and the UV spectrum changed across sunrise and sunset. And so we did a whole series of measurements for them, and you can see that here the sun is above the horizon and here the sun is below the horizon, and you get this change in ratio of the M and the UV opsins. So we can link all this back to the circadian work as well. Thank you very much for listening, and I'm happy to answer any questions either in the chat or at the end.
Thank you, Anne. That was fantastic. And yes, I think that any discussion on daylight is not complete without considering the ultraviolet radiation as well. So we're going to move on to our last talk for this evening before the Q&A. And this talk is from Phil Butlin from the University of Edinburgh. So the screen is yours, Phil.
Okay, great. Can you see that okay?
Yeah, that was great. Thank you.
Just trying to move that out of the way. Okay, great. Yeah, just thank you for that, Paul. And I'd just like to say before I start, thank you for the Daylight Academy for inviting me to speak. I think everyone's talks have been really interesting so far, and I'm really looking forward to the Q&A at the end as well. So my research is slightly different from everyone else's in the fact that I'm a plant molecular biologist as my predominant aspect of my work. And I study what's known as the shade avoidance responses in plants. So that's in relation to how plants can get information from their light environment and how they change their growth in response to this. So I'm going to be talking a bit about the research background to that, and also then relating it to how daylight measurements can inform us about different types of responses that might happen in nature and what aspects of daylight measurement are particularly important for us. So I'm aware that most of the people who are watching probably aren't plant biologists, but I should hope that you all know that at least that light is fundamentally important for plant growth. And this is particularly because of photosynthesis, which is the process by which plants derive energy using light from the sun in order to produce sugars, which feed themselves, and consequently it's how they feed all of us as well. But the impact of light on plant growth can go beyond this, as plants also use changes in different characteristics of light, such as its intensity, its spectral composition, its period length, and also its direction or angle of incidence, as ways of sense or getting a sense of where they exist in the world, both spatially and temporally. So for instance, plants can use changes in day length relative to internal molecular rhythms in order to determine what season they're in and consequently whether it's time to flower. And in addition, plants can monitor changes in spectral composition of light in their environment in order to kind of build up an idea about the 3D structure of the environment that they've germinate in. So for example, when plants grow in open environments, they receive largely unfiltered sunlight, which will have a spectra that looks something a little like this. However, as light passes through heavy canopy cover, such as in a wooded area, we tend to see reductions in intensity and also this dramatic shift in the spectral composition of light. And This is partly because of the role of chlorophyll, which is the predominantly used photosynthetic pigment used by plants. So chlorophyll preferentially absorbs, or most effectively absorbs, wavelengths in the blue and the red regions of the visible spectrum. And as a result, plant material heavily absorbs blue and red light, whereas other wavelengths, such as those in the green and also the far red regions of the spectrum, aren't absorbed, and instead they pass through or can even be reflected off of plants. And this will then lead to these drastic changes in spectra. And plants are able to sense these and use this information to determine whether they're in an open, light abundant environment or in a shaded, light scarce one. So what does this all mean for plants? Well, if you're like me and you have lots of plants in your home, you'll be well aware of the fact that they can be very sensitive to their environment. And unfortunately, whereas houseplants are lucky enough to have someone to simply move them to another location if they're not thriving, plants in nature are unfortunately confined to the environment in which they germinate. And this can obviously pose an issue to plants, as not every environment is going to be perfectly suited to their needs. So to try and combat this, plants have evolved various forms of developmental plasticity, which is the ability to change their development in an attempt to adapt to their environment. And a classic example of this involves, well, if you imagine a plant which germinates in a heavily in a forest environment, so it's on a forest floor and there's very low levels of light, which will obviously reduce its photosynthetic outputs. Some plants have evolved to thrive and tolerate these environments, whereas others display these changes which essentially help them to negate the negative impacts of them on their growth. And collectively, these changes constitute what we term the shade avoidance syndrome. And this is what we study in the Halliday lab at the University of Edinburgh. So the shade avoidance syndrome is typically characterized by these elongations in plant architecture. And these changes are thought to help enable plants to essentially forage for improved light by growing out of the shade. So in the model organism Arabidopsis, which is what we work in, we tend to see stem elongation in seedlings, as well as leaf stem elongation at a cost of reduced leaf blade area in adult plants. And we also see these upward leaf movements, as well as accelerating flowering. And the extent of these changes on plants' development can really be highlighted by watching them growing in these two videos here. So on the left, we've got wild type plant showing normal growth. And on the right, we've got what's called a shade constitutive mutant. So it's essentially, it can't turn these responses off. And this really just demonstrates just how extensive these changes can have, of an impact these changes can have on plant development. So how do plants tell when they're in shade? Well, we know that plants are able to sense various wavelengths in various regions of the visible and near visible spectrum. And they do this using certain different families of photoreceptors. So for example, plants can sense light in the blue, red, far red, and also the UVB parts of the spectrum. However, whilst all of these are able to be affected by changes in shading, we know that of particular importance to plants are changing the amounts of red and far red light. And this is because, as I already mentioned, red light is heavily absorbed by plant material, whereas far red isn't. And this will make shade environments relatively far red enriched compared to these open ones. And plants are able to sense this and tell and determine what we call the red to far red ratio. So this is the comparison between the amount of red and far red light in an environment. And they do this using a family of photoreceptors known as the phytochromes. So phytochromes are sensitive to red light in their inactive state, which causes them to become active. And in contrast, their exposure to far red light in their active form will inactivate the phytochrome. And this means that they have red far red reversibility. And as a result of this, their activity is greatest in open environments where there's a high ratio of red to far red light, whereas their activity is lowest in environments where there's such as shade environments where we've got low red to far red ratios. So once activated, phytochromes serve to suppress the molecular level processes which drive these elongation responses. And therefore inactivation under shade relieves this suppression and triggers these elongation growth. So because plants then use red to far red ratios as a reliable indicator of external shading, we also use them in our daylight measurements as a way of getting a sense of whether a plant might be considered an environment shaded or not. So physiological studies in lab settings have enabled us to decipher several key characteristics of these processes, including how they're shaped by changes in rate-to-fired ratios, and also how giving signals at different times of day can alter the strength of the response. But unfortunately, the need to work under controlled conditions in order to decipher between or distinguish between true and confounding effects has led us to use largely artificial conditions in labs. And these often fail to capture some of the subtle complexities of natural environments. And this has only been emphasized by, or amplified by the fact that we don't really have access to these the technology to more accurately recreate these conditions. So for instance, in growth chambers, it's often either continuous light regimes are used where we don't ever turn the light off, or where we have square wave light regimes where we have sharp light dark transitions at dawn and dusk and fairly steady light levels throughout the day. But in nature, we'll actually see spectral, see irradiance outputs would look something like this, where we have fluctuations throughout the day and more gradual light dark transitions at the extremes of the photoperiod. And in addition, whereas in nature, obviously, the sun's position will change throughout the day and thus change the angle of incidence and the direction of light. In growth chambers, we use lights which are fixed directly above plants, so these remain static. And they also emit these light with a spectra which isn't really representative of a normal daylight scenario. And recently, several papers have been published which have suggested that actually under these artificial conditions, plants may respond and grow slightly differently. So for example, the authors of this paper, of the Panther et al. Paper, showed that the plant's circadian clock may function differently under these natural fluctuating conditions. And this can obviously be an issue because the plant circadian clock is known to regulate virtually all developmental processes in plants, and it's including the shade avoidance responses, unfortunately. In addition, work from the Halliday lab in Edinburgh has shown that one of the 5 phytochromes in Arabidopsis, known as phytochrome A, actually may accumulate and function differently under gradual light-dark transitions, which is again highlighting this the fact that what these types of responses we see under artificial conditions may be not truly accurate of what happens outside. And unfortunately, as the focus of these responses has shifted more towards understanding the molecular mechanism in recent times, we've partly lost the connection between what we see in experiments and what happens outside. So this is where our interest in daylight measurement comes into play, as when we're collecting, understanding what happens in these natural environments is vital for shaping our knowledge of these developmental responses. So when we collect daylight measurements, there's several factors that we need to consider. And this includes how changes in fluence rate, changes in the rate-to-fired ratio, but also broader spectral changes such as those in blue, green, and UVB irradiance. And therefore, any equipment that we use to take these measurements really needs to be able to factor in and collect data relating to all of these facets. And this is also particularly important because these responses are, well, not only because of the high complexity of these light environments, but also due to the complexity of the responses themselves. So for instance, we know that several different types of shade scenarios can occur in nature. For example, under forest cover, we tend to see deep shade environments occurring, which is where we get low fluence rate and low rate-to-fired ratios simultaneously. In addition, another scenario can occur where we get high reflectance of far-red light off of neighboring but not directly shading plants. And this can lead to more moderate reductions in rate-to-fired ratios and create what we term a neighbor or proximity shade environment. And interestingly, these authors have, and a couple of other papers have shown that the responses in Arabidopsis seedlings may be slightly different to these two types of environments, with seedling elongation actually being significantly more suppressed under these deep shade environments compared to the proximity shade ones. And interestingly, it's even been shown that the location of where shade signals are perceived by plants can also alter the type of response that's exhibited. So these authors demonstrated both in the model organism Arabidopsis and in black mustard plants that when a shade signal is perceived on a leaf stem as if being shaded from above, it will actually cause that individual leaf to elongate out as if it's to enable it to grow back into the light, essentially. Whereas when far-red signals are perceived at the leaf tip, as if from an approaching neighbor, it will cause the leaf angle to increase and therefore avoid it that way. And then when we see shading given to the whole of the plant, we tend to see a combination of these two responses occurring. And these two studies together really highlight the incredible adaptability of these processes and the fact that considering these as a simple on-off mechanism driven by whether there's far red present or not isn't really sufficient in trying to understand these processes, because really we need to begin to consider factors beyond just these spectral changes on how plants respond to these conditions. So really, it can become incredibly difficult to try and unpick all of the processes at play here. And this is particularly due to the fact that we're trying to characterize a response in plants which is regulated by a multitude of intersecting and overlapping factors. And the reality is that every plant will face a unique set of environmental restrictions, which will in turn face shape a unique developmental strategy. And really we'll quite often see conditions not occurring independently of one another in nature. So for example, in addition to low red to far red conditions, it's also known that low blue light conditions can also trigger elongation in plants. And this is interesting because we'll often see both of these conditions occurring simultaneously in these deep shade environments. And this could then in turn be altering what type of response we see. In addition, we also know that high temperatures also trigger elongation growth. And this has actually been shown to also work through the shade-sensitive phytochromes, which is also highlighting the overlap between multiple different responses in plants. So really, we'll see combinations of all of these conditions occurring in nature. And it really isn't sufficient to consider to only look at how plants respond to one signal at a time in these experiments. But unfortunately, we don't really know very much about when or where these specific overlapping conditions occur in natural settings. And this is partly due to the fact that there hasn't really been much environmental daylight measurements being collected in relation to these responses. And that's something that we think really needs to be improved in the field in the future. So just to summarize, daylight measurement is really important for developing our knowledge of these processes. And this is particularly because of the continually growing set of information which is indicating that these processes can be regulated by more factors just than beyond just the red to far red ratio changes. So traditionally, whereas we've considered red to far red ratios as a reliable indicator of shading, it actually appears that these responses may be driven by more factors than just this. And this is because of their complexity, which has largely been evolved to try and cope with these equally complex environments. So really, daylight measurements for us need to begin to go beyond the current methodology and start to incorporate how other factors, such as changes in the direction and the angle of incidence and the time of day and season, as well as also looking at how weather can alter the spectral composition of light in these shade environments. And this will then begin to develop our, I guess, our perception of the ecological relevance of these responses in nature. So then finally, beyond just the fundamental side of this work, it also has immense agricultural implications, because often we see these responses being initiated in dense cropping environments, which lead to poor resource allocation and also eventually crop loss due to lodging, which is essentially where plants over-elongate and their stems break. So if we can potentially reduce these responses in crops, we'll be able to improve farming yields and outputs. And this really highlights some of the potential impacts that this work can have on agriculture. So I'd just like to say thank you for listening. I look for your questions and the field spectroscopy facility, guys.
That was brilliant. Thank you, Phil. Really interesting talk. So that takes us to the end of the talks, and I'm just going to share this again with you. So reminder, questions in the Q&A tab. And so I'm going to start with a question for Anne. So the UV radiation measurement project was quite a big international undertaking, I would imagine. What does it take to get a collaboration like that off the ground in respect to daylight measurement? Oh, I think you're muted, Anne.
We were very fortunate in that we had this sort of global impetus of the ozone hole and ozone depletion causing this sudden interest in UV around the world. I mean, everybody just wanted to, we've got to measure the UV. And so that was the driving impetus. And it was just as the EU was starting to provide funding for work across the whole European Union. And so those two things combined just made it we were very fortunate to sort of jump on both those bandwagons, if you like, and get a whole series of EU funded projects. Without that funding, I don't know how it would have worked. I mean, by the time we got to the intercomparison in 1997, we got people from Canada and New Zealand saying, can we come at our own expense and put our instruments alongside yours? So it'd become truly global at that point. How you get that going without a sort of boost, I don't know, but I would suggest the Daylight Academy is a really good place to start. People measure daylight for so many reasons, but having that system where you can easily compare from one, in our case, one place to another, or from one glass house to somebody's desk to somewhere else, is really important.
Yeah, that's really interesting. Thanks. So I'm going to turn to the questions from the Q&A chat box. So the question at the top here is from you and Edie, and I'll open this up to everyone. So the question is, what is your one go-to piece of daylight measurement equipment? Maybe start with Brian.
Portable spectrometers and more Grzelski pyranometers of various kinds with shadow bands to take out the direct, so you can measure the diffuse and direct separately. So a combination of instruments. Sorry,
not just
one.
instrument.
Not just one.
All
Sorry. Well, yeah.
Manuel?
I think I have two go-to measurement equipment pieces. One is a photo research PR670 spectroradiometer, which is really, really nice and high spec and provides really, really high quality data, which is the moon measurement I showed in the talk was made with this, and it really provides nice resolution. It's not portable, right? I think it comes with a battery, but you have to, basically it's a relatively big device. And I have, I think for kind of field measurements, a smaller portable ocean optic spectrometer, which is quite nice and USB driven and very, very convenient to use. The problem with that is, of course, then a little bit, if you wanted to do measurements that you ran continuously, you can't easily take dark measurements, right? There's no shutter that would enable you to just turn it off. So you have to somehow, either do it manually or come up with a parametric model to discount the dark.
Yeah, I think we might have the same one. Phil, any thoughts?
Yeah, so we basically, one of the classic for us was using USB Ocean Optics USB 2000. That was really great, and that's been used in our lab for years, and that's always worked really well. However, one thing that we did find was that it needing to be plugged into a laptop was kind of limiting in the sense of us trying to take measurements in the field in Scotland isn't really ideal for that, as any water is going to pose an issue. So we've also got a more portable LICOR, I think it's called Li-180 spectrometer, which I think is designed for sort of more use in greenhouses, but it actually works really well because it's super portable and produces really good data for us.
Great. And do you have any thoughts as well? What's your go-to piece of measurement equipment?
I think I gave it away in the talk. I use a Bentham double monochromator scanning spectroradiometer. for the sort of climatological measurements and also in the lab. But like Manuel's, it's not really portable. I'd say it's transportable. We have it in a big case, and it's called a coffin. It's about that size, so it's all temperature controlled and everything. But for sort of quick work, I also have an ocean optics spectroradiometer and have used that. Again, we put it in a box, and it lived outside up in the Arctic for several weeks, so they can do that if you put them in a nice environment box. In the UV, you do have to be a little bit careful at the shortest wavelengths. They do struggle there, but across the rest of the spectrum, great. And for human work, dosimeters.
Yeah, so I think that actually touches on an interesting point, because most of what was mentioned was obtaining spectral data. And as was mentioned in the talks, that can tend to be maybe a bit more complex or a bit more expensive to carry out. So maybe Manuel, if you wanted to comment on how we can make daylight measurements a bit more accessible.
I mean, I think it comes down to this question of what is the information that you want to capture. And I mean, I think if you coming back to this idea that daylight is statistically relatively regular, and basically you can capture it within a reasonably low number of dimensions, I think then in principle it should be reasonably straightforward to estimate daylight spectrum from three responses, right, from RGB responses. I think in practice, the reason that doesn't work is that we're not actually exposed to the illuminant spectrum, right? We are exposed to different stuff that reflects and absorbs light. So the actual dose that we kind of want to measure isn't necessarily daylight, but it is daylight interacting with all the various pieces of material and stuff that we have in the environment. And so I think that's where kind of these approaches kind of breaks down. Ultimately, I think what we care about, I mean, coming just to the circadian field, I think we now have reasonably good evidence that melanopsin drives most of the responses that we care about over the timescales that are important. It doesn't mean that cones and rods don't contribute. I think there's still reason to believe that there might be cone and rod contributions. But for the large scale effects, I think you probably would be, it might be sufficient to have a melanoptic sensor that's reasonably well spec'd and is matched in its spectral sensitivity to the melanopsin spectral sensitivity. And I think it also then boils down to really, if you had spectral measurements, well, ultimately, to determine something like dose, you will anyway project it into, I mean, for circadian response, you project it into the alpha-opic irradiances. I think there's a use case for having spectra, which is when you're thinking about, is it in any way, shape, or form useful to know what type of spectrum someone was exposed to? For example, if we realize, oh, they're mostly exposed to some specific lighting, well, maybe that's a target for intervention. But I don't think spectral identification per se is the physiologically relevant variable.
Okay, that's interesting, thanks. So the next question on the list is also addressed to Manuel, but I think I'd like to get Anne's opinion on this as well, maybe after Manuel gives his opinion. So, and there's actually two questions I can see that are similar. So can you explain further how to determine the spectral diet over 24 hours? What devices are needed? And are these continuous measurements over 24 hours? And the related question is, can you identify a simple and easily understood way of representing the daylight diet or daylight exposure over time?
I mean, I think, to me at least, this concept of the spectral diet is more a hypothetical device, because I think that, I mean, it basically is the time course of spectral irradiances that we are exposed to. I don't think that the spectral diet per se is, as I kind of said before, is the physiologically determining variable. But I think it is something that certainly should be something that's measurable. I think in practice, I mean, there's now various devices that have been developed by research groups that are able to either measure alpha-opic radiances or irradiances or spectra that are, basically measured in the corneal plane or on a lapel or something like that. I think these come much closer to the quantity that we'd really like to know, which is not so much corneal irradiance, but actually retinal irradiance, right? In a way, the problem is that the only instrument that allows us to determine retinal irradiance is the retina, and we can't put a sensor in there or read it out. So I think that's a little bit the challenge. That even if we had small devices that were in the right plane, I think we still would factor in retinal, the fact that all the light goes through the pupil is filtered, etc. I think in terms of metrics, I mean, I think one thing that I find quite appealing is the idea of looking at basically the time above threshold. So having defining specific thresholds, and I think this is an approach that I've seen formalized by a paper by Catherine Reed a couple of years ago, where basically you ask, okay, what is the amount of time spent above a specific threshold? What's the timing of that time spent above a specific threshold? And what's the intensity? So you capture a lot of things with this type of metric, intensity, duration, and also the timing in the 24-hour cycle. I think that's an approach that I think seems very promising.
Great. And maybe Anne, from your perspective with UV radiation, think about the spectral diet of how much do we need, how much is too much, and how can we tell?
That's if you've got a few hours. Yeah. Okay, so I'm going to come at this from the human skin and talk about the good and the bad, the erythema and the vitamin D. Both instigated by UV radiation, and the bottom line is, to get your vitamin D, you do not need to get sunburn. So never sunburn is the bottom line. Don't do it. And most of the measurements that are made refer to something called the SED, the standard erythemal dose, or the MED, the minimal erythemal dose. The standard erythemal dose is 100 joules per meter squared of radiation that's been weighted with that erythema action spectrum. The minimal erythema dose is the amount of erythemally weighted radiation that it takes to give you, so it's a personal measure, you a slight sunburn, just ever so faintly pink. Okay, and so we know that people with very fair skins will start to go pink at about 2 SED. And if you've got dark skin, if you're Afro-Caribbean black skin, then it's many times that, and you're not going to sunburn for a long time. So that's used a lot in public health. And it's kind of, because it's almost a safety measure, no one wants to sunburn, even when we're not talking about sunburn per se. It's often used as a sort of safety measure, and it's linked to that UV index that I told you about, so that you can go and get a measure of it from the weather forecast, in actual fact.
Great, thanks. So we are fast running out of time, so I'll move on to another question. I'll address this one to Brian, maybe. So can one imagine a metric that would be relevant and used in all disciplines to enhance interdisciplinary studies? So I'm thinking of your giant matrix of daylight measurement properties.
Yeah, I think the simple answer is no, because you've got, as we discussed, quite different responses in different systems. And I think one of the things is to be able to translate measurements from one discipline to another and know how to relate them. As Anne said, there's the data that goes with the measurement. Are we looking at raw data? How is that processed? What's the angle? What else is measured, etc. So that it is, what plane is it measured on? So that it actually is translatable between disciplines. So and the examples there, for example, you've got environments where you've got both plants and people. So you've got quite different responses there, but you want to use the same input data to relate to those things, to relate, for example, physiological studies of human health and how the brain responds to light, to the actual light environment, as Manuel said, in buildings, which is not, daylight, when you think about it from an architectural view standpoint, is the light you get from daylight coming into buildings reflected off surfaces. It's referred to daylight, but actually it is a much more complicated phenomena, and it's generally reflected off surfaces. And well daylit buildings are all actually about surface design, surface color. Okay, so I think it's about how you adequately translate it rather than looking for a universal measurement, because I think that could again lead to well, it would lead to suboptimal solutions ultimately.
Yeah. Okay. So I'm going to ask two more questions, and I'm going to break my own rules and go right to the bottom of the Q&A because I want to hear from Phil again. So there was a question here, which I think has just disappeared. So maybe Phil has just answered it.
I just answered it.
Okay. Yeah, maybe you'd like to just talk about that a little bit.
So the question was, I'll just find it. So the question was, how is the influence of light linked to other factors like nutrient and water content of the soil and temperature? And is there a complex interplay here, and how can this be measured? And essentially, we do know that there is a lot of interplay between these pathways, particularly at the molecular level. So for instance, a lot of the signaling proteins which are involved in coordinating these responses are actually common between multiple pathways and do lots of similar responses to different conditions. So we know that there is a lot of interplay, but it's really difficult to actually measure this and quantify it, or even just to study it, because if you want to look at how the effects of one factor are regulating plant growth, you kind of need to sort of eliminate all of the possible candidates that could change these responses. So, for example, when we look at the effects of light on plant growth, we tend to not test other factors, or try and eliminate other factors such as how wind is involved or how temperature. So we tend to use consistent temperatures because these will all play a role in shaping these responses. But there is some really interesting papers which have shown, for example, links between, and I did link one in the answer to the question, which have shown connections between different cropping environments, so different levels of or different, far irradiances in crop environments, and changes in drought tolerance and nutrient resistance and things like that. And it's definitely an emerging field, but it's something that is, it's quite difficult to really unpick at this point.
Yeah, that's really interesting. So I think the last question, and I'm going to open this up to everyone, and we'll maybe come to Manuel first. So, and this was actually touched upon in Anne's presentation as well, that we have a lot of people that measure daylight and different reporting levels on daylight as well. So different techniques, different equipment, and different quality of reporting on that as well. So how do we promote good practice in daylight measurement? And is this something that we could do through the DLA?
I mean, I think this is a really, really good point. I think ultimately, to make measurements comparable and really harness the power of having different groups of researchers collecting data at different sites under very different contexts just requires an approach to harmonize data. And I think it's really essential that we come to some type of framework that defines at least minimum variables to be defined, measurement type, whether it was calibrated, what type of quantity was reported, and really agree, I'd say, on a set of pieces of information that are necessary for a data set to be considered according to some specific standard. I think the Daylight Academy would be a great vehicle for this, and I think it's definitely much needed. Otherwise, I think the problem is we kind of disappear in this data, in this flood of data that are collected, but there's no real way to compare them, aggregate them, and harmonize them. I think there's probably some systematic ways of going about doing this. So there's methods to generate expert consensus, and I think that's certainly something that I think the Daylight Academy could really help facilitate.
So Phil, you're sort of in your PhD now. What sort of resources would you like to see, or have you seen that have sort of helped with promoting the good practice in daylight measurement?
That's really well. I think actually, so we've definitely. Since starting my PhD, I think the exposure to the Daylight Academy stuff has been really useful because I've kind of become more and more aware of the fact that there is this big community out there of people who are all trying to measure similar things. I think we've even had meetings before to discuss possibilities of doing stuff like this, and I think that really is so useful for me because, as I say, my research is more molecular biology and less related to actual spectral measurements. So actually having that resource sharing and understanding from and knowledge sharing from other people has been so helpful. And I think for me as well, like knowing more about what technology people have and what equipment might be open for sharing, or for even just using data sets from, that can all really, I think, really help expand our sort of knowledge and application in these fields.
Yeah, those are some really good points. And Anne, any lessons on good daylight practice from your experience with these sort of big collaborative measurement networks?
I think that once you get an enthusiastic group of people together who just want to make things work, then it will happen. You know, it needs somebody driving it, I think. But there's clearly a lot of enthusiasm out there and a lot of interest in this. So I think it just needs somebody to pick up the baton and run with it.
Great. And maybe Brian, you could have the final word.
Thank you very much. Yeah, I mean, fundamentally, I mean, it's not the fault of anybody, but daylight existed before science existed, and different subjects evolved with their own context and measure the things that were relevant to their subject. We now can look at much more systematically, and underpinning that, the ability to handle greater amount of data, greater data resolution, share data is there as well. So we're at the time where this is possible. And whilst there is a relevant of devices that are, as I said in the presentation, are essentially weighted to particular parts of the spectrum, maybe a particular angular response or whatever, in a particular, their valid application, how they are calibrated and perhaps also perhaps with very low, as the cost of portable spectrometers decreases, having additional spectrometric measurements so that things become comparable, and then maybe developing, because standards were developed in the context of a particular application. So there will be a standard for measuring daylight in buildings, which is to do with the visual response in bright light, essentially. Getting more general standards that are actually about how you make measurements translatable, and I think that is important, and is now, I think, possible with portable equipment, possible with data sets and the kind of tools that Manuel described. So I think that, my final thing is there's a project within a thing called the Daylight House within the Daylight Academy, which is going to be to try and bring together metrics and measurements and that's available to participate in. So as a driver for exactly that kind of activity. And what I'd like as a maybe a parting plea is to bring together people from the broadest range of disciplines as we have today, so that there are, because I think the potential insight is also the question, why does that discipline measure that? And could that be relevant to my discipline? And for example, the behavior of shading in buildings is not that different from the behavior of canopy in a forest. Plants are responding to that, people respond to something else. Why are different things measured? Are there some things we can learn just from the fact that certain things give certain responses? Perhaps we should measure that as well. And I leave that as a final parting thought.
Great. So it is possible, and maybe the Daylight Academy will be one of the pioneers in getting this to become a reality. So with that, I'd like to thank all the speakers for contributing towards this seminar. It's been really insightful. I think I've learned a lot as well, and there's a lot of questions to think about moving forwards. So before we go, I'd like to just bring your attention to Daylight Awareness Week, which is next month. 3 reasons why we need daylight. Please do come back, listen to these really interesting talks, and engage with the Daylight Academy. And with that, I'll thank you for listening. Thank you very much, and hopefully we'll see you soon.